So good afternoon, ladies and gentlemen. I'd like to introduce myself. My name is Jennifer MAYKL, and my teammates are here. Anna BATMANOVA.
Hello.
And Kajal KARKI.
Mayan.
And today we're gonna explain to you our presentation, which is the degradation of plastic polymers using genetically modified bacteria. So why did we choose this project? We wanted to find a sustainable method to reduce plastic pollution in Luxembourgish rivers and rivers worldwide. Plastic pollution is known as the accumulation of synthetic polymers in the environment, where they cause problems in wildlife and their habitats, as well as for human populations.
Plastics are non-biodegradable, meaning it'll take them hundreds and thousands of years to be broken down in nature, which then causes an accumulation of plastics in wastelands. So with the project, we want to find a sustainable method to speed up this process of degradation and so reduce plastic pollution.
Another reason for doing this project is that during my internship week at University of Luxembourg in biology department, I saw how much plastic was used every day. After that, I discussed this with my friends about the increasing use of plastics in our daily lives, which encouraged all of us to find a solution of this increasing plastic crisis. And after doing some research, we then come up with a few interesting ideas.
So this is a proposed method. Unfortunately, it is all only theoretical. We couldn't carry out this experiment due to COVID-19 restrictions.
The first step of our method was to insert a foreign DNA into our vector Escherichia coli, also known as E. coli. Vector is a means of delivering genes into a cell using gene technology. The reason for using E. coli is that firstly, it is easy to access. Secondly, it is cheap, and most of the strains of E. coli are humans. Then Escherichia coli transcribes the gene on foreign DNA that codes for the enzyme.
The bacteria then translates this code to produce the amino acids, and then multiple amino acids come together to form our enzymes.
We then tested how these enzymes break down plastic, specifically PET.
The summary of our method. So we assumed that we would start off with only one strand of DNA and E. coli bacteria. The method we use to multiply our DNA to a big enough sample is called polymerase chain reaction, also known as PCR. This is a type of technique commonly used in forensic sciences. To modify the bacteria, we use the restriction enzyme to cut the part of plasmid of E. coli. And DNA ligase is used to link together sugar phosphate backbone of the DNA molecule, coding for PETase and METase and the plasmid, which produces a closed circle of double-stranded DNA.
The next step is to insert the modified plasmid into the E. coli. This is done by adding concentrated calcium ion solution to the bacterial cultures. The bacterial cultures are then given a heat shock to make sure that the plasmid passes through the cell surface membrane. The next step is the identification of the modified E. coli. The initial DNA coding for PETase and METase is given a genetic marker, specifically GFP extracted from jellyfish. GFP fluorescence when exposed to UV light. So our expectation is that the E. coli bacteria will fluorescence when it's exposed to UV light, which tells us that it has been taken up by the modified plasmid. Hence, the uptake of the bacteria by modified plasmid is proportional to the intensity of fluorescence when exposed to UV.
Once UV light was shown onto the bacteria, some of them would have been luminescent because if the gene was taken up, then the luminescent protein could have been produced, which is what we detected. The luminescent marker was used instead of an antibiotic resistance marker because the results were easier to visualize and the whole process would have been less cumbersome. So then the next step would have been to clone the bacteria that has successfully taken up the plasmid. We achieved this by separating the luminescent bacteria from the non-luminescent ones and then giving them time to reproduce by binary fission. Binary fission is a type of asexual reproduction by the separation of the body into two new bodies, where each new organism has a copy of the original DNA. We would then introduce our PET plastic into the culture and leave the bacteria to degrade it. The results were recorded by determining the concentration of CO2 gas produced. We chose this method of measurement as microbial degradation of a long-chain polymers, such as PET, involves the release of CO2 gas. So the concentration of CO2 gas produced is proportional to the amount of plastic broken down.
So these are our theoretical results. We state that the bacterial cultures containing PETs and METs only modified bacteria will break down the least amount of plastic and have have a really low turnover rate. The bacterial culture containing PETs and METs break down the most amount of plastic because the enzymes work in a two-enzyme system.
As with every project, there's also going to be some advantages and disadvantages. So our project aims to use microorganisms to provide a solution to the problem of plastic pollution, save energy, as well as achieve to have cleaner oceans. Today, most of the plastic is incinerated once it has been used, and so useful material and energy is lost. So by the use of bacteria, this energy does not have to be used up, and then materials can still be available.
Unfortunately, there are some disadvantages too. Our modified bacteria is only used for PET because the enzyme that we propose is very specific. We assume that the process is going to be slow as the turnover rate of the enzyme is very low.
So looking into the future, we'd actually want to carry out our experiment and make further discoveries to hopefully reduce the problem of plastic solution. And as stated before, we'd also want to find different solutions for different types of plastic other than PET.
We would also ideally want to modify our enzymes PETase and MHTase to have a higher turnover rate so that they degrade plastics faster and so make the whole process of plastic degradation more efficient.
And finally, we want to apply our experiment here in Luxembourg and measure the plastic content in the Luxembourgish rivers and lakes. And hopefully we can degrade the plastic pollution here in Luxembourg with our experiment. Thank you for listening.
Thank you for listening.
Thank you very much for the presentation. Any questions?
Yes, I have a question. Thank you for your report. How did you estimate the level of degradation that you showed us a few slides ago?
By concentration of carbon dioxide being produced.
But your work is theoretical, right?
Yes.
And how did you estimate it theoretically? Can you explain, please?
We couldn't do those calculations ourselves. What we only had was theoretical information from research papers, which showed how each enzyme worked.
Okay, I understand. Thank you.
Thank you for your presentation. Can you tell me, do you have any ideas on how to increase the effectiveness of this enzyme and how to influence the rate of the reaction?
I'm sorry, could you repeat your question once again?
Yes. So do you have any ideas on how to increase the effectiveness of this enzyme and how to increase the rate of the reaction?
So as stated before, we said that we would be giving the culture a heat shock. So this basically activates the enzymes to produce a higher turnover rate. So the heat shock corresponds to the turnover rate initially.